Okay, so this video is about non-disjunction. So when we look at meiosis one, in metaphase one, we have homologous pairs line up along the metaphase plate, and then as we go into anaphase, those homologous pairs separate. But sometimes the homologous pairs may fail to separate. So look at that bottom left homologous pair, and as we go through anaphase one, look how the homologous pairs stay together. Now, as those cells finish meiosis, you can see how that results in some gametes having an extra chromosome over here on the right, and some gametes missing a chromosome over here on the left. But not so that process of failure to separate is the non-disjunction. So they're staying together. Now, non-disjunction, though, can also happen in meiosis 2. So here we have metaphase 2, where the duplicated chromosomes line up along the middle of the cell. Now, I want you to watch this bottom left cell. So here, sister chromatids fail to separate during anaphase 2. So then as meiosis finishes, now half the gametes have an abnormal number of chromosomes, where the other half are fine. So the two on the right over here are normal, but because the sister chromatids failed to separate in anaphase 2, you end up with one gamete that has an extra chromosome and one gamete that is missing a chromosome. Now, when we define this, we call having an extra or missing a chromosome, we call this cell aneuploid. Aneuploid, the AN means not good. So when we define this, having only one of a particular chromosome is called a monosomy, which I may be pronouncing that wrong. So here, these cells would be missing one chromosome, so they would have only one. I'm sorry, when they fertilize an egg, that zygote would only have one of a particular chromosome and not two. And then in trisomy is where they have an extra copy of a chromosome. So like here, carrying this extra copy would lead to an individual that had three of a particular chromosome. Okay, so when we look at how this could happen, for example, so maybe a sperm non-disjunction in the process led to the formation of that sperm. So now here, 2n equals 45, which is our normal 46 minus 1, 2n minus 1. Or this could happen where maybe a non-disjunction, maybe the egg had an extra chromosome. So here that would lead to 47 chromosomes, 2n plus 1. Now, on this slide, I mentioned, I typed, I want to point out this is not the same as haploid. This is having like only missing one chromosome, not an entire set. We're talking about a single chromosome missing. Okay, so let's go ahead and look at some examples of karyotypes showing non-disjunction. So here's a normal karyotype. We can see we have two of all of our autosomal chromosomes here and two sex chromosomes, an X and a Y. So 2n equals 46 here. Now, if we were like as we just mentioned, monosomy is where you are missing just one chromosome. So maybe like here, one copy of this autosomal chromosome was missing. So that would be monosomy. I'm sorry, I don't know. Now, generally speaking, though, human embryos that are missing a copy of any autosomal chromosome will fail to develop during birth. So sometimes if a female experiences a miscarriage, it could be that maybe it's the result of non-disjunction and missing one of the chromosomes. And if we remember, our chromosomes are what carry our genes, and then it's our genes that code for proteins. So by missing a whole chromosome, it may lead to too low a dosage of a particular chromosome. I'm sorry, too low a dosage of a particular protein, which leads to errors during development. Now, what can be survivable, though, in some cases, is having an extra chromosome. Now, having, like in Down syndrome, for example, having this extra number 21 does lead to extra protein products, which do lead to an imbalance during development. And that is why people with Down syndrome will have different developmental issues because of that imbalance of proteins produced. Now, you could also have trisomy 18 or trisomy 13. Those are also survivable. Now, what we notice in these cases are these chromosomes are small chromosomes. So having a duplicate or extra copy with the extra proteins can have some issues, but it's still survivable compared to maybe having an extra number 1, which is our largest chromosome. That may not be survivable. That imbalance would be too great during development. So sex chromosomes, though, are a little bit different. It's a little bit easier to survive having an extra or missing sex chromosome. So here, having 2 copies of the Y chromosome is called Jacob syndrome. And the Y chromosome only has about 30 genes. So that's a bit survivable. We have an example of Turner syndrome, though, where a female is missing an X. This happens in, I think, 1 out of 5,000 live births for females, and this will lead to the female being sterile and some other developmental issues. Okay, now one last point I want to make is that aneuploidy, though, is not the same thing as polyploidy. So poly means many, and ploidy or ploid kind of means like sets or numbers of chromosomes. You can think of like haploid as 1 set, diploid 2 sets, triploid 3 sets, etc. So in polyploidy, this is where an organism may have more than 2 sets. So as humans, we are diploids, but here we have a karyotype of a triploid where it has 3 of every type of chromosome. Strawberries are actually octoploid where they'd have 8 of each type of chromosome. So here I just took a human karyotype and added a 3rd, but generally speaking, polyploidy would be more common in plants, for example, and that's partly because plants can self-fertilize, so their own abnormal gametes can self-fertilize. So anyway, we can talk more about polyploidy in our speciation topic on how new species evolve. Okay, so that is my discussion on non-disjunction. Great job.